Hey folks, in this video we cover the next section of chapter 4, all about synapses. And on this opening page I have a picture of an actual synapse under an electron microscope, and you can see that there's a bit of a gap here in between two bigger structures. One here, this is going to be before the synapse or presynaptic, and this is going to be after the synapse, postsynaptic. These arrows right here are pointing at something called synaptic vesicles. And those vesicles are full of neurotransmitters. You can see that they're dark here. That's because they're full of neurotransmitters. You can see little dots in some of these. Those are actual neurotransmitters like dopamine or something like that. You can see they're hanging out at the synapse and they're going to cross over to the other side. So we're going to walk through what's going on at the synapse once we've reached the end of this neuron in these slides. So as I mentioned, synapse just means a gap, right? So it's nothing, right? It's the space in between two neurons or two structures, maybe a neuron and the muscle, for example. And so it turns out that neurons don't directly touch each other all the time, and there's this gap in between. Most of these synapses are going to be between one neuron and the dendrites of another one. Okay, so the dendrites hanging off the soma typically are going to receive signals from the axon, the terminal of the axon of another neuron. There's a technical name for that is axodendritic. And so the dendritic shafts and then the spines on those shafts, they're like buds on the branches of the dendrites, are going to pick up the signal from across that gap. Now, to be clear, there are other synapses in your brain. They can go directly to the cell body, they can be internal, and then they can go axon to axon technically without going through dendrites. But most of the time we're going to be talking about this model, and that's what we're going to cover here. To put this in the broader picture, we've been kind of walking through the action of the neuron from the setting up the resting potential to receiving postsynaptic potentials. That's part one here, right? So we're sitting at rest and we get postsynaptic potentials from one neuron to another. This is going to be a synapse here. Okay? Those postsynaptic potentials are going to come on down and get summed through temporal summation and spatial summation. You remember this, right? And if we reach enough excitatory potential to reach threshold, then we'll fire an action potential, right? We're going to reach threshold, we're going to fire an action potential, and that's then going to be conducted down the axon. The myelin sheath is going to facilitate that, right? To help that action potential be conducted more quickly and efficiently. We're going to reach the terminal button. At this terminal button, we're going to have something called exocytosis. We'll talk about that in a second, but that's going to continue the pattern where these terminal buttons are now going to typically connect to another neuron's dendrites, and we'll start the sequence over again. So let's zoom in on that synapse, right? Between the terminal button of one neuron, that we would call presynaptic before the synapse, and then the dendrite and the dendritic bulb of dendritic spine of the postsynaptic membrane. There's a little gap here as they connect. And this connection gets maintained over time, so it doesn't really move around much, but they're sort of facilitating this connection chemically. You're going to see we've got, again, this is a cell, so it's going to have things like mitochondria and Golgi complex, that sort of thing. But then you'll also remember that we have these things called microtubules running up and down the axon. Synaptic vesicles that are going to be holding neurotransmitters. You can see the little dots in there. Those are neurotransmitters. Okay. Those synaptic vesicles are either going to be carried down through microtubules, or they're going to be, some of them are going to be made down here. And then they're going to be hanging out down at this synapse. And the goal of the synapse is to get these neurotransmitters out of the presynaptic neuron and to connect to receptors on the other side. So as mentioned on the last slide, these neurotransmitters are going to get created, they're going to get synthesized, and they're going to be packed into synaptic vesicles. Okay? This happens in two different places. Some neurotransmitters are made just right at the synapse. This is typical of small neurotransmitters like glutamate, GABA, even monoamines like dopamine and serotonin and norepinephrine. Small neurotransmitters typically just get made right down there at the synapse itself. They get packaged into vesicles so that they can be held like in a package that can get released. And they do it right at the terminal buttons. Larger molecules, like neuropeptides, oxytocin, endorphins, that sort of thing, get made at the cell itself and the cell body, the soma itself, and then they get carried down those microtubules. One consequence of this is that a single neuron can actually make small neurotransmitters at the terminal buttons and also make larger neurotransmitters at the soma. And so a single neuron could release more than one kind of neurotransmitter. So that's not always the case, but it can happen. Okay, so we're going to make small ones down right at the terminal buttons, ready to go. We're going to make bigger ones up at the cell body, transport them with microtubules. So now we've got neurotransmitters down at the terminal buttons, right? They're sitting presynaptically and ready to go to be released and go postsynaptically so we can send a neurotransmitter to send a signal, right? So how does that work? How do we release neurotransmitter molecules? Well, this is all going to be based on that action potential, right? The whole reason we set up the resting potential was so that we could get action potentials. And the whole reason we have action potentials running down the neuron, running down the axon, is so that we can release neurotransmitters. Okay? So we're going to run the action potential down. Remember, this is activating, exciting, and then sodium is going to be rushing in as we go, and we're going to get these waves of excitation as we go down the axon. At the very end of the axon, we're not going to have sodium channels anymore. We're actually going to have calcium channels. Okay, so it's just a different mechanism, but it's going to work the same way. Once we get high voltage coming in, those calcium channels are going to open, and the calcium channels are going to interact with the synaptic vesicles and with the presynaptic membrane to release. Okay? And that's going to be called exocytosis. Technical term here. Exo means like exit, going out, and then cytosis is like death of a cell or a structure, right? So we're breaking the synaptic vesicles, cytosis, and then that's causing the exit of the contents of those synaptic vesicles. So once the synaptic vesicles get released, the neurotransmitters are then going to be hanging out in the synapse itself. Okay, so that's the whole point of the action potential. The whole reason we set up the resting potential got us an action potential. The whole reason for the action potential is to then cause exocytosis so that you can get dopamine or glutamate or whatever it might be into the synapse itself. You can see a figure here of exocytosis. Here's an electron microscope of a real synapse, and you can see presynaptic synaptic vesicles up here. Some of them have been released. You can see the synaptic vesicle here is empty. That's because these little dots, these neurotransmitters, have been released into the synapse, and that means that now the contents, the neurotransmitters, can come and interact postsynaptically. And here's just a blown up sort of cartoon version of that as the neurotransmitters get released. Now, what happens on the other side? Let's say I release glutamate or dopamine or serotonin or acetylcholine into the synapse. Well, there's two general categories of effects you can have on the other side. The neurotransmitter is going to bounce around and then is going to activate and interact with receptors on the other side. And these work like a lock and key. So a certain neurotransmitter fits in a certain receptor. So if I release multiple kinds of neurotransmitters, only one kind can attach here. So let's say this is glutamate, right? This receptor will only respond to glutamate. Okay, so we got these specialized receptors on the other side, and they have two general types of effects. The first type of effect is called ionotropic, which is like the word ion, that are just going to lead to EPSPs or IPSPs on the other side. Okay, so I attach a neurotransmitter here, and then it opens the channel, and I get either an excitatory response or an inhibitory response. Okay, so maybe the neurotransmitter opens, and then I get a rush of sodium over here that excites the neuron. Or sometimes this is open already and the neurotransmitter can come in and close it, so I can then inhibit an existing response by having maybe less um, sodium action. Okay, so this is the basic mechanism we were talking about earlier, where you get little bursts of excitation or inhibition. Those get summed up on the neuron on the other side. So these are ionotropic receptors. There are other types of receptors that don't just excite or inhibit, but that lead to longer term changes. And there's a lot of complexity in how that works. But this figure kind of breaks it down, right? Where the receptor comes in and it has a downstream effect, right? So instead of just opening up a channel so I get excitation or inhibition, we're going to like release proteins on the other side that are going to change the neuron on the other side. So this might do things like helping learning, right? So if I'm consistently activating two neurons together, those neurons you might have heard neurons that fire together wire together. Well, this is going to be due to metabotropic changes. Okay, so if I activate this metabotropic receptor on the other side, it might make that neuron easier to activate later on by causing changes to the way it works. There's a third type of receptor that's not postsynaptic. Okay, so the neurotransmitters are running around. They're gonna the main role is going to be activating the postsynaptic receptors, so I can get EPSP or IPSP or longer term changes. But they'll also sometimes connect presynaptically, essentially to provide feedback. Okay, so we got a lot of glutamate going on here. If there's so much glutamate, some of it's going to activate presynaptically, so I can get feedback on what's going on. That might sort of slow down exocytosis or speed it up. The final step in the phase is, well, what do we do with all those neurotransmitters, right? So I spent all this time releasing something like glutamate on the other side so it can have an ionotropic effect on the other side. Well, now I've got a bunch of glutamate in the synapse. I need a mechanism for dealing with that released neurotransmitter. There's two ways of doing it. The first way is called reuptake, where we bring the neurotransmitters back into the presynaptic neuron. You see a figure of that here, right? So exocytosis releases the neurotransmitter. It's going to activate receptors on the other side, and then it will be taken back up, reuptake, through a transporter on the other side. Okay, so that's reuptake. The other mechanism is called enzymatic deactivation or enzymatic degradation, where the enzyme deactivates or degrades the neurotransmitter. So break it down into its parts so that you can recycle it and either use it here or elsewhere. One example of this is acetylcholinesterase, which is an enzyme, and it gets its name, acetylcholine esterase, because it breaks down acetylcholine. Okay, so acetylcholine gets broken down into components, and then we can use those components, acetate and choline, elsewhere in our body, elsewhere in our brain. Okay, so between one of these two mechanisms, we're not going to let those neurotransmitters go to waste. We'll either recycle them through reuptake or through enzymatic degradation. As with everything in your brain, there's some complexity and there are some exceptions. So most neural communication is going to happen through synapses and through neurotransmitters. But there are some cases where a neuron will just connect directly to another neuron with a very small gap, and then you'll just have a junction between them, and the action potential can basically just continue. Okay, so the action potential from one side can then continue to the next neuron and just continue. We don't need any neurotransmitters in these electrical synapses. We can just have a wave of excitation just like we did down one neuron. This happens in a couple of specific places. First, in those astrocytes that we talked about, which are glia. So glia don't have neurotransmitters, um, but they can make connections. So astrocytes are those like star-shaped glia that coordinate activity with all of their arms, right? So those have gap junctions, and they can just pull in activation to coordinate without using neurotransmitters. The other time this happens is in developing brains. So before our brains form all of the synapses and prune out synapses that don't work during early development, um, those neurons are going to connect together and have gap junction activation between one neuron and the other one. Eventually, those synapses are going to form, and we won't use the gap junctions anymore, and we'll start using neurotransmitters. But early on, early developing brains are just going to use a simpler gap junction without all the neurotransmitter stuff going on. Okay, and that's it for the synapse. Next time we'll do a quick walkthrough of different kinds of neurotransmitters and how they work.